Welcome everyone to the Fermentation Association's webinar of Our Fermented Foods Probiotics. I'm Amelia Nielsen-Stoll, editor of TFA. We are a trade group that was launched to support producers who use fermentation to create delicious and often healthful food and beverages. Our goals are to help educate consumers about fermentation and its benefits, support scientific research into those health benefits, and work with food safety authorities to establish clearer and more appropriate regulations in regards to fermentation. Today, we bring you two great speakers, Dr. Mary Ellen Spencer, Executive Science Officer of the International Scientific Association of Probiotics and Prebiotics, and Dr. Maria Marco, Professor of Food Science and Technology at University of California, Davis. We have many questions already submitted and reviewed with our speakers. If there are additional topics you'd like to see addressed, please enter them in the chat below and we will try to get to them. I will come back at the end to answer some of the additional questions in the chat, but for now, I will turn it over to you, Maria and Mary Ellen.
Thanks, Amelia. So it's my pleasure to introduce Dr. Mary Ellen Sanders, who's a consultant in the area of probiotic microbiology. So Dr. Sanders received her bachelor's degree in food science from the University of California at Davis. She then went on to receive her master's and PhD degrees in food science from the North Carolina State University. For over the past 30 years, Dr. Sanders has worked with international food and supplement companies to develop new probiotic products and to offer perspective on scientific substantiation of probiotic product label claims. A few notable activities from her career include her efforts serving on GRAS determination panels and participating in the working group convened by the FAO, WHO, that developed guidelines for probiotics. Dr. Sanders is the current chair of the United States Pharmacopoeia's Probiotics Expert Panel. She serves on the World Gastroenterology Organization Guidelines Committee, preparing practice guidelines for the use of probiotics and prebiotics by gastroenterologists. Mary Ellen is also collaborating on clinical studies to validate probiotic safety and efficacy. These activities have resulted in over 130 peer-reviewed publications in microbiology, efficacy, substantiation, and regulatory issues, mostly pertaining to probiotics. Last but not least, Dr. Sanders is the founding president of the International Scientific Association for Probiotics and Prebiotics, or ISAPP for short, and currently serves as its executive science officer. As a member of the ISAPP board of directors myself, I cannot overstate Mary Ellen's contributions over the years to the success of ISAPP, an organization that is dedicated to advancing science, promoting evidence-based clinical use, and improving consumer understanding of probiotics, prebiotics, and fermented foods. So with that, I'll turn the mic over to you, Mary Ellen.
Thank you so much, Maria. And I really appreciate everybody signing on today to share this topic. I'm hoping to be able to provide perspectives that are going to be useful for you for differentiating between probiotics and fermented foods. So let me share my screen and get started here. So the first thing I want to do is point out that my email address is here, maryellen@isappscience.org. So if any of the questions or you have any follow-up that I wasn't able to address, feel free to reach out to me after this webinar. And again, another important component of my talk is to just make it clear what organizations I've worked with. So I have several for-profit organizations, companies that I have consulted with, as well as some nonprofits as well. So those are my disclosures. So Maria suggested that I provide just a bit about ISAPP before getting into the meat of my topic today. So ISAPP, or the International Scientific Association for Probiotics or Prebiotics, is a nonprofit organization founded in 2002. It's governed by an all-academic voting board of directors. And as Maria stated, its mission is to advance the scientific excellence of probiotics and prebiotics. And over the past 5 or so years, we have expanded our reach to include fermented foods, postbiotics, and symbiotics. We're supported by member companies, and we have a very active student and fellows association. And I'm going to just take a moment and talk about some of the consumer-facing outputs that we have that might be of interest to you. We have 16 different infographics, and many of these are available in different languages, and they are very, they're full of just facts about these particular topic areas. We have 6 different videos. They're all 3 minutes or so in length, and we have subtitles in a variety of languages too. And we have a very interesting blog program where we have board members and guest experts that will submit perspectives on different timely issues in the related to this field. And then also we have a monthly newsletter that goes out to the full ISAPP community, probably close to about 5,000 people now. And it's very simple to sign up for the newsletter if you might be interested in what the organization is doing. We do convene annual meetings. Typically our meetings are closed registration, although we are hoping to have at least a component of our 2021 meeting as a virtual open registration component. So that hopefully will come in June. We do a variety of consensus panels, and I'm going to speak to a couple of those during my presentation today. We publish a lot of state of the science and opinion papers in peer-reviewed journals. So just check out our website to see the list of those, and most of them are open access. We collaborate with other organizations and comment on regulatory and scientific papers, and we have a challenge of trying to increase our international outreach. So those are all things we're focused on now. And I do want to call your attention to a really exciting project we have right now, and it is going to be coming out hopefully within a week or so, certainly by mid-March, a continuing education course that's targeted toward dietitians, and it will be published by the Today's Dietitian platform. And this particular course is focused on evidence-based use of probiotics, prebiotics, and fermented foods for digestive health. And it's free to access, whether or not you need continuing education credits. It's really going to, it's just full of very interesting and useful information about this area. So with that introduction into ISAPP, let me, and there's our website, www.isappscience.org. So go there. So let me jump into our topic now. So we're talking about fermented foods and probiotics and how they relate to each other. So I wanted to start just with a definition. I know that everyone on the call here knows what a fermented food is, but this is a consensus panel definition that was just published earlier this year. And it's foods made through the desired microbial growth and enzymatic conversions of food components. And let's contrast that to probiotics. Probiotics are live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. So you can see that probiotics and fermented foods have very different definitions. But let's look a little bit more closely in terms of how probiotics and fermented foods differ. Now, this graphic was taken out of one of the ISAPP infographics, and I'll call your attention to these 2 columns here. The first one is probiotics, and the second is probiotic fermented foods, which is essentially a fermented food that does contain a probiotic. Now, both of these, you'll notice, have the exact same responses to each of these criteria. So these, we don't make any distinction between those 2 with regard to these probiotic criteria. But let's go through them, because there's certainly difference between those 2 categories and fermented foods. So first of all, what about a documented health effect? Well, certainly probiotics and probiotic fermented foods must have evidence of a documented health benefit. Fermented foods, the definition does not require that. How about microbes alive at the time of consumption? Probiotics absolutely have that as a requirement. Fermented foods do not have it as a requirement, and they may or may not have live microbes at the time of consumption. What about microbes being present at levels that provide a health benefit? Again, probiotics must have that as a criterion. Fermented foods are not required to have that. Safety for intended use, of course, is required across the board. Are the microbes taxonomically defined to the strain level? And for probiotics, they must be. For fermented foods, there's not a requirement for that. And finally, with regard to the genomic sequence of the microbes that are contained in the product, probiotics require that, and fermented foods do not. And so in short, there are quite a few differences between probiotics and fermented foods, and you cannot assume that a fermented food is a probiotic food, even if it has live cultures present. And further, if you are interested in recommending health benefits from a fermented food in an evidence-based manner, Many traditional fermented foods fall short. They don't have the controlled randomized trials that will provide a causal link between the food and the health benefit. So keep in mind, a food may be nutritious, but in order to meet the criteria of a probiotic, to have a probiotic benefit, the health benefit must stem from the live microbe, not just the nutritional composition of the food. Otherwise, you just have a nutritious food that happens to have like live microorganisms in it. You don't have a probiotic food. But let's talk a little bit more about what the sufficient bar for evidence is. I mentioned already randomized controlled trials, but I think it's interesting to look at the paper here that was recently published that delineates the criteria that qualify microorganisms as probiotics in foods and dietary supplements. And what this paper states is that what a probiotic needs is that it must be supported by at least one positive human clinical trial conducted according to generally accepted scientific standards. And typically that means a randomized controlled trial. Now, I will say that this is a minimum requirement, and there are those in the probiotic field who would say that perhaps one study is not enough, that you really should have a minimum of 2 studies, that one that confirms the results that were found in the other study. And this would especially be true for medical uses. If you were developing a probiotic, for example, for a therapeutic use, you would want more than one study. Now, if we look at the type of evidence that's available for probiotics, what we find is that there are quite a few randomized controlled trials for different strains on different endpoints, and that enables you to evaluate the strength of the evidence for any number of different conditions. And I just have shown here several meta-analyses that have been published, some of which are published by the Cochrane Library, which is just the highest level of quality in terms of meta-analyses, and also one meta-analysis that was conducted by the European Society for Pediatric Gastroenterology, Hepatology, and Nutrition, or ESPGHAN. And so the point with this slide is that there's lots of studies that can be pulled together and considered for strength of evidence for different endpoints. So if we look at a representation of the hierarchy of types of scientific information that's available, that's shown here. And I just wanted to show you where the probiotic evidence falls on this hierarchy. Now, this hierarchy goes from the bottom of the pyramid, which really is scientific evidence that is challenged with having a high risk of bias. In other words, these are expert opinions that really have not been controlled in a way that would support, that the bias isn't managed in a study such as these. But as you go up the pyramid, your risk of bias starts to drop. And certainly at the top of the pyramid, if you have a properly designed randomized controlled trial, that can be done with a very low risk of bias. And further, if there are several of these trials that have been conducted on similar endpoints with similar interventions, you can pull them together into a systematic review and look overall at the quality of the evidence. And this is the type of evidence that is available for probiotics. Now, the probiotic field, it's true, does have many randomized controlled trials, not all of which have a very low risk of bias associated with them due to flaws in how the trials were set up. But for the most part, there are many randomized controlled trials and systematic reviews that support the evidence for health benefits for probiotics. Now, if we look at the evidence that exists for traditional fermented foods, that evidence more centers in this area with case-controlled studies and cohort studies. And the difference, the reason that this evidence is considered to be higher risk of bias is because you don't have the ability to causally link confidently the intervention, or in this case, the fermented food, with the health endpoint. You know, you might have good evidence from these studies that a health endpoint is associated with consumption of a fermented food, but you don't know that it's necessarily causes the health benefit. So the level of confidence that you have in the relationship between consumption of a food and a health benefit increases, and you want to have the low risk of bias studies conducted in this way. And fermented foods typically fall short of that. So observational studies suggest that consumption of some fermented foods is associated with improved health outcomes. But as I said, there typically are few randomized controlled trials. Now, having said that, there are some randomized controlled trials, and this slide just shows a few of the non-dairy randomized controlled trials that are done with traditional fermented foods, and these are RCTs. So these would be able to, if properly done, indicate a causal relationship between consumption of these foods and the health benefit they were looking for. So one of the issues, besides evidence, is that you need to have, for probiotics, clear strain-specific identity of the microbes. And so we've already discussed the need to have specific low risk of bias studies that show and document a health benefit. Another criterion is that a probiotic must be well defined in terms of the strain that's used as the probiotic. And one of the ways of doing that is by characterizing these individual probiotic strains with a high quality genomic sequence with an annotation. And then further, probiotic strains should be named properly, bearing the genus, the species, and subspecies that conform to the current nomenclature for the particular organism. Another point about strain-specific identity for probiotics is that you have to have stipulation of the strain designation for each strain in the product. And so I didn't mention this at the start, but a probiotic always should be labeled with a genus, a species, a subspecies if it's called for, as well as a strain designation. And without that strain designation, you really can't tie the evidence that may be published on that strain to the product. So it's really important that that strain designation be carried on product labels. That strain designation should be used in all publications. So it's very clear the evidence that's been published can be linked to a particular probiotic that's in a product. And another really important aspect of understanding very clearly the live microorganism or microorganisms that are in your product is that it allows for consistent manufacture of the same product. And then that allows you to repeat studies appropriately. If you don't have a good definition of the intervention of the food and the composition of the food that you're using in an intervention trial, then you're not able to really properly repeat that trial to see if in fact the same result is obtained. So unfortunately, many fermented foods do have a microbiological content that doesn't meet this standard. So there are many fermented foods that do not have microbes that have the genomic sequence known, where the microbes that are present are properly named at the genus, species, and strain level. And so for that reason, it's another difference between probiotics and many traditional fermented foods. Now, another challenge and difference between these between fermented foods and probiotics has to do with the quantities of live microbes that are present at consumption. Now, this is a table that's taken from a paper out of Bob Hutkin's lab from a couple of years ago. And what he did, and he and his group did, was they looked at the levels of colony-forming units of live microbes that have been published in a variety of different fermented foods. And the point I want to make with this is look at the very wide range of colony-forming units that might be present in different fermented foods. And contrast that to a probiotic supplement, where you have a very clear indication of the number of microbes that are present in the product. So this probiotic supplement is appropriately labeled. It has 12 billion CFU, and that's the number of live microbes that should be available at the time of consumption or at the end of shelf life. So a probiotic has a known effective dose, and whereas fermented foods very often contain unknown levels of microbes, and especially at time of consumption. And so that's another key difference between many fermented foods and probiotics. Now, people may argue with me and say that I'm just being too picky or too demanding in terms of me insisting that probiotics are not the same as fermented foods. And one argument that could be made would be along the lines of there are many stellar, top-notch scientists studying the human microbiome who repeatedly assert that ingesting fermented food delivers live microbes that may help your gut microbiome and your immune system. And so why isn't that enough evidence? If these high level scientists are saying that these fermented foods may deliver these benefits, isn't that enough to be confident of a health benefit? And it's interesting because a very nice paper that I saw recently published by the Sonnenbergs talked about the microbiota, the current status of industrialized people and their current microbiota, and what that means with regard to various disease outcomes. And this is a quote taken right from the abstract of that paper. And what they said was that rapid modernization, including medical practices and dietary changes, is causing progressive deterioration of the microbiota. And so they start out with that as a baseline understanding about the current status of our microbiota. But then they go on to say, as we hypothesize that this may contribute to various diseases that are prevalent in industrialized societies. And then they go on in the paper to further hypothesize that consumption of fermented foods and also fibers, but for our intents and purposes, fermented foods may help reverse or manage the situation with increased diseases. But I just want to point out that they acknowledge this is a hypothesis. And a hypothesis is not the same as a fact, a proven fact. And so although it's a very, very interesting evolving area of research, and studies such as these do a great job providing strong support for hypotheses, a hypothesis hasn't yet been proven. Oops. And so I just want to point out that well, let me conclude after this slide. So I wanted to add this slide is to just show support for the idea, for the hypothesis that was expressed in the Sonnenberg paper, which says that, which has to do with a project that has been initiated by ISAPP. And what we are doing is we fully agree that it's a very interesting hypothesis that consumption of live microbes may in fact lead to health benefits. And we want to explore to see what kind of evidence might strengthen that hypothesis. And so what we've embarked on is a review of the NHANES database to estimate the live microorganisms that are associated in the foods that Americans eat and looking at associated health outcomes. Now, again, look at the word associated. It's not proof that if what we find in this study shows that people who eat higher levels of live microbes have lower health risks or better overall health, that doesn't prove that the live microbes caused that, but it's another piece of information that can help us down the path of trying to get that proof. So we certainly agree with the hypothesis and are trying to explore that ourselves. But the point I'm making here is that a strong hypothesis is not the same as proof. And evidence for probiotics are required to meet a higher standard than associative studies, many of which are tracking biomarkers and not health endpoints. So with that argument, I would say, no, it's not convincing enough to be able to say that probiotics and fermented foods should be considered equivalent. Another argument goes something like this, that, look, it's really obvious when you search on the internet or even when you go into certain scientific publications, you very often will read headlines that say the best way to get probiotics is by eating fermented foods. And here's just one example of many such articles that you'll find. This article was titled 11 Probiotic Foods That Are Super Healthy. And then when you look at their list is essentially all fermented foods. And so I would, of course, say that this list is not an appropriate list of probiotic foods. And then, of course, I would go on to say the obvious, which is just because someone points it out on the internet doesn't make it true. And so unfortunately, there's a lot of misinformation of this type that's available on the internet. And not only is it available on the internet, but sometimes it's available in publications that are put out by highly reputable organizations. And so this is a brochure. It's a 17-page brochure put out by the Harvard Medical School that talks about the benefits of probiotics. And throughout this publication, it conflates the probiotics and fermented foods. And in one form or another, talks about fermented foods being the best source of probiotics. And so again, even the experts who come from highly reputable organizations can miss the subtlety of this issue. Now, for the rest of, or for the next several slides, what I'm hoping to do is to cover a topic that is a bit of a nuanced topic. So I hope I can explain this well. But I think it's an important concept for this audience because it can provide a framework for understanding why we have a tendency, why we are so driven to generalize from the microorganisms that compose fermented foods and generalize to health benefits that they may impart. And this idea comes from a couple of, we discussed it in a couple of ISAPP publications. We introduced this concept of core mechanisms that may drive health benefits in the Hill et al. Consensus panel paper on probiotics. And then we developed it a little bit further in a 2017 paper that's shown here. But let me walk you through what this is, what basically comprises this concept. So first of all, I need to start out and just remind the audience that when we talk about probiotic benefits, we correctly consider them to be strain specific. And I mentioned this already, but essentially you have a genus, a species, and a specific strain of that species that's been tested and shown to have a health benefit. But it is, and these individual strains, though, when you look at the composition of those strains, when you look at the genes they harbor and the mechanisms that they express, oftentimes you see among a group of strains of the same species shared mechanisms. And what this figure shows is that in some cases, they're widespread shared mechanisms across many, many strains of the same species. And so that may, for example, an obvious one would be the ability to produce lactic acid and other short chain fatty acids, and usually in situ. And so what you see is that this might be a mechanism that many, many different strains have. And the presence of that mechanism may in fact drive certain important benefits, such as competitive exclusion of pathogens, which then may lead to decreased incidence of certain infections. And so there are many of these mechanisms that we hypothesized might be widespread, some others that might be frequent, but not quite as widespread as others, such as the ability to synthesize vitamins, maybe the ability to make certain enzymes that can lead to certain benefits. And then there are probably mechanisms that are very much strain specific, and you wouldn't expect to find these mechanisms expressed across many different species, across many different strains of the same species. But when you consider the fact that there are widespread mechanisms, when enough strains of the same species are shown to have a mechanism that's widely shared, as well as demonstrate a benefit that results from that mechanism, and here's the key to this concept, it may be justified to consider that that benefit is species specific, as opposed to only unique to a specific strain. Now, there's an example of this. There aren't many examples, but there is one example of this exact situation. And it has to do with the ability of yogurt cultures to improve the digestion of lactose by lactose intolerant people. Now, I will say that the European Food Safety Authority actually had a positive opinion, essentially agreeing with, from what I know, is the only positive opinion on a probiotic health claim in Europe, which is the relationship between these yogurt cultures and improved digestion of lactose. But what's really important about this EFSA opinion is that this opinion was expressed at the species level. It was tied to the Streptococcus thermophilus and Lactobacillus bulgaricus starter cultures that are present in essentially all yogurts. It was not tied to specific strains of Thermophilus and bulgaricus. And that's the important distinction I want to make here, is that the enzyme involved in promoting the digestion of lactose in humans, that enzyme is present almost always in strains of these species. And for that reason, it's reasonable to extrapolate the benefit to other strains of these species. And so that's an example of a core benefit delivered by a mechanism that's shared among strains of a species. Now, when the consensus panel got together in 2014, or I guess they got together in 2013, but when they got together and discussed this issue, they said that if you did have well-conducted human studies that document a health benefit on different strains of a specific species, you could justify that, you could use that evidence to support the benefit at the species level. And if you were able to do that, you could use the description contains probiotics, because this evidence exists that strongly suggests that you would expect that benefit to be delivered by all strains of a particular species. But this evidence, these human studies, these well-conducted human studies are the baseline for that. And in fact, those well-conducted human studies were available for the claim on lactose intolerance in yogurt cultures. So the panel did say that the term contains probiotics could be useful. However, they said that there would be, that they did not believe that you should put specific health claims on the label, that you would need strain and dose specific information for a specific health claim. And then further criteria would include proof that sufficient viable microbes are delivered in the product, and that you would need proper identity to the strain level of the microbe. So those are other criteria that are, as I've already discussed, that are important for probiotics. So I'm hoping that that concept of core benefits came through okay. So as I wrap up this webinar, I really wanted to conclude with an analogy. And since I am a horse lover and I own a horse, I thought I would use my horse Noah to try to show an analogy for this issue of fermented foods and probiotics, and how important it is to use the proper words and the proper descriptors. Now, Noah is a very dear horse to me. I've had him for 18 years and we've been good companions the whole time. He has a lot of positive qualities. He's really good with kids. He's a sure-footed trail horse. And in his day, he was a good little jumper. So he has very many benefits. But as great as this horse is, and I'd be the first to tell you how great he is, he's not a thoroughbred. He has no papers saying he's a thoroughbred. And when I tell people about Noah, I don't describe him as a thoroughbred because I know that the word thoroughbred has a specific meaning and I should respect that meaning. And I feel that sometimes the communications about probiotics and fermented foods with composition of probiotics, that they miss the point that it's important to use the proper names and descriptors for the food so that there's not a chance of misleading people about what the nature of the particular product is. So in summary, I'm a huge fan of fermented foods. I did not spend any time going into the value of fermented foods. I know that all of you guys are well aware of those. My goal here is to encourage proper communication about them and about probiotics. So I strongly believe we should adhere to accepted definitions. The probiotic definition is a widely accepted one. It has been around for almost 20 years, and it was reaffirmed 6 years ago by a consensus panel of experts. And the definition requires live microorganisms that have been shown to be associated with have been shown to confer a health benefit and that are delivered in sufficient quantities in the final product. If the word probiotic isn't used properly, then it is going to completely lose its usefulness and it won't mean anything to anybody. So I think it's really important to use the word properly. Now, I recognize there may be a cachet in using the term probiotic, but only probiotics that meet the criteria that I described in this webinar should use it. Now, descriptors such as contains live active cultures, or this is a fermented food with live microbes, can be used, and those are suitably descriptive, but they can be used if the criteria for correct use of the probiotic is not met. So I'll go ahead and stop there, Maria, and turn it back over to you.
Thank you, Mary Ellen. Amazing presentation, really informative. And I'm excited to ask you a few questions now about your talk. One point that really hit home for me was, the necessity to actually study these foods or probiotics in people, so in randomized controlled trials. And I know this is something you and I have talked about, but I'd like to just kind of come back to that. What are some of the challenges that need to be overcome, or at least acknowledged, in our randomized controlled trials on fermented foods?
Yeah, I think one big one is blinding the trial. So we talked a little bit about making sure that you keep the risk of bias in any study very low, so that you can have confidence that you have a causal relationship between your intervention and the health benefit. You don't want to introduce bias. And one great way to not introduce bias is to blind your study, so that the clinical coordinators or the people who are involved in the study don't somehow influence the way that the people consuming the product might describe their health outcomes associated with that. So blinding is critical. But blinding, if you're doing a study about pills, is very easy. You can take a pill and you can put just, inert material in one pill and the active material in another, and that's very simple to do. Blinding foods is not, because the level of microbes, the composition of the microbes that are in the product have a lot to do with what that product tastes like and what it looks like. Mm-hmm. And so the placebo, or the control that's used for these randomized trials, you want to be able to blind, which means that no one would be able to tell the difference between the two, the control and the active. But of course, with foods, that can be a real problem, because it's just hard to make those foods without having the right, without having the live microbes.
Yeah, that might be something we just need to acknowledge in these studies. And there was a question that came in, about the financial aspects and how costly these studies can be. But could there be a pathway where we could study these foods for health benefits, not just at a product specific level, but at higher levels, like categories of foods?
Yeah. I mean, that's a great question. And that's why I wanted to bring up this idea of core mechanisms, because I understand that these trials are very expensive to do. But I also understand that if you look into the literature base, you can see many, many, many of these trials have been conducted. And I'm certainly not saying everyone is trying to do it the right way, but there are many people in the probiotic field who are trying to do it right. And they launch, large, well-controlled intervention trials, and they are very expensive to do. If you don't have the money to do that, I think that you have to consider, or I would prefer that you consider using a descriptor other than probiotic for it, so that you don't imply that your product has evidence that it doesn't have. But now, with the idea of a category level type of study, and Maria, this is where a lot of the research that you do really could come into play, is can you identify mechanisms that might drive the health benefits? Can you develop that hypothesis? And that can be done in pre-clinical types of studies that are done in the laboratory. So if you can get a sense that a mechanism can drive a health benefit, you can identify that mechanism in a category of food, then yeah, you probably could consider developing the rationale that you could do one study showing the benefit and then try to make the link that because that mechanism is present in what was studied, that it should also confer a benefit for other members of that category. Does that make sense?
Yeah, and that's one thing that really has me excited about this concept of core benefits. And again, it is still in many respects a hypothesis for many fermented foods. And the yogurt example really resonates because it is a very specific enzyme, very specific property of yogurt that's conferred by many strains of those species needed to make yogurt. So that I think is kind of opens the doors to possibility on fermented fruits and vegetables, where there's certain microorganisms that we'd expect to be there. But again, we need to know really if how they're healthy, or if those microorganisms are needed to make those foods healthy. So here's a particular scenario that has come up. Say we have a fermented food where a known probiotic strain has been added into it.
I knew this was going to come up.
This is where we're going. But we've talked about this over the years and thinking about matrix effects and, would it be necessary to have another human study if a defined probiotic strain was added to a fermented food?
Yeah, no, it's a great question. And it's one that a lot of people way smarter than I am have grappled with. I think there's two camps of thinking with regard to that question. The first camp is that probiotics can be viewed as an ingredient to foods. And just like you can take vitamin D and add it to any food, and you don't need to redo your study with vitamin D in every given food you make with it, you can consider that the benefits are associated with the strain. And if you take that strain and deliver it alive at the proper levels, that you have a reasonable expectation of delivering that same benefit. And that ingredient approach is used quite commonly in the industry. There's another camp, and it's a reasonable challenge to that, is that, hey, these microorganisms, the probiotic organisms, are alive, and they are active. They can change, they can evolve, they can respond to their environment. And because of that, you can't necessarily assume that if you put a live microbe in one environment that it's going to necessarily interact or behave in the exact same way that it did in a different one. And I think there's value to both ways of looking at this with regard to, and I think at the end of the day, what you have to be able to do is think through the individual circumstances with regard to the product you have and make a reasonable scientific assessment of why you would think that benefit should be retained in the product that you are formulating with it. I don't think it's okay, in my opinion, to just throw probiotics in whatever and just say, yeah, it's going to be fine. But I do think that if you look at available evidence, there's evidence to suggest that probiotics have a certain level of resiliency or elasticity that allows them to function to confer certain benefits in different matrices. And then we also have to recognize that we eat food. And so as soon as you eat you could have a probiotic capsule. As soon as you eat it, it's going to be mixed with all the foods that are already in your stomach. And so it's going to have those environmental challenges immediately anyway. And so, but I just think you have to spend some time thinking about it and just making sure that you're not just, coming to a conclusion because it's a convenient conclusion.
Mm-hmm. Yeah, I agree. Just a couple more questions before I turn it over to Amelia. Kind of on the other end of that spectrum, though, where we have a lot of fermented fruits and vegetables, for example, that may contain live microbes, but we don't know their strain level, there's no known recorded health benefit for that food or that product. You mentioned this idea of live microbes or contains live cultures. Is that do you see that as a sort of a way forward where there's sort of a compromise on the use of the term probiotic, which is kind of recognized as being live microbes present?
Right. Yeah.
Is that an alternative?
In a way, that when I see a fermented food that says probiotics on it, I very often think what they're trying to communicate on that label contains live microbes. I think that that's what they're trying to communicate, because I'm doubting, at least with some of the products I see, that they have any evidence of a health benefit. And so they're just looking for a catchy single word that will communicate to people this has got live microbes in it. Well, in my opinion, that's a misuse of a word, and that shouldn't be done. But I'm not a marketing person, but it seems that the concept of live active cultures is something that resonates with people as well. So why not use that? Or why not even just say live microbes or live microorganisms, live bacteria, or some version of that. And that's correct, assuming that it is correct, assuming it's not pasteurized after fermentation, but and then if it is, maybe you say it's made with live cultures.
Right. Yeah, thanks for clarifying on that. So my last question has two parts, and of course, so you've been working on probiotics for 30 something years. And so you really bring a unique perspective to the situation where we are today. And so I was wondering, how have you seen probiotics field change over the years? And the second part of that is, what do you see coming in the future?
Yeah. The field has grown so much. When I first got into the field back in 1990, there were just so few randomized controlled trials. There were just a few that were being done in Europe, and one or two of them in the United States. There just was very little body of evidence, and it was a field that was propelled by great hypotheses. And you fast forward, I haven't looked on PubMed lately to see how many, hundreds of clinical trials have been conducted, but there are many, many trials. And so that whole body of clinical research, I think, has really makes the difference between where it used to be when I started out and where it is now. Because now there are certain endpoints where you can say there's reasonably convincing, actionable evidence to say that probiotics should be expected to have some benefits. But I think that there is an incredibly long way to go. We don't understand in many cases the mechanisms behind the benefits. I think it's reasonable to say, because these benefits are conferred by live microorganisms, it's probably more than just a single benefit. A single mechanism. So more than just one enzyme that results in it. It might be, a whole variety of different mechanisms that come to play to lead to a particular benefit. So it's going to be quite complicated to sort it all out. Having said that, I think that the juxtaposition with all the incredible research on the microbiome now has really catapulted the whole idea of probiotics forward. And so I think that people now recognize how important microbes are to human health, and probiotics are just an obvious way to mediate those, potentially leverage a live microbe to impact positively the live microbes that we harbor. And I just think that that's an incredible development in the field. And I think that what we're seeing now and what's going to be propelling us into the future are very specific strains that are developed for very specific functions. And I think many of these will come out in the drug category as we go forward, that they're going to be therapeutic purposes. And I think that we're going to, we're going to see a lot happen in this field over the next 5-10 years.
Thank you. I'm really excited for that. Okay, Amelia, you are going to field some questions?
Hi, yes. Dr. Sanders, this has been fascinating, very enlightening. Thank you so much for that presentation. So we had quite a few audience questions about what will happen with the FDA adopting this definition. We had, let me read this question we had Dr. Perez-Diaz, hi, from the USDA, she asked, what do you see are the next few facts that are needed to be researched to move forward in achieving an official probiotic food designation in the US?
Well, that presumes that we should have a probiotic food designation legally recognized in the US. And I think that is a function of, or that's something that could be discussed. You know, from my point of view, the science is clear. The parameters that have been established by experts in the field, I think, are pretty well laid out. If the regulatory, if regulators are interested in defining a category, I think it would probably be fairly straightforward to do. You know, you just have to sort through, how are you going to enforce it, or how are you going to make sure that the term is properly used? So right now, my whole argument is based on a scientific argument that these words have meanings, and that those meanings should be respected. If you want to take this to the level of government enforcement, that is a whole new level, but I think that the foundation is already there to make that happen. So if you'd like to discuss it, I'd be more than happy to.
We had a question from David from Sonoma Brinery. He asked, using wild strains of lactobacillus for fermented products, and with the uncertainty as to exact strain and CFU count, it seems the few of the fermented foods sold today actually should use the term probiotic. What are your thoughts? Do you agree?
Yeah, I mean, yeah, I think that's probably true. There are some fermented milks, some yogurts that I know have been studied in randomized controlled trials as and shown to be confer a health benefit, and the probiotic strain is properly characterized, and all those meet the definition. But I think many of the traditional fermented foods likely do not. But I have not surveyed the marketplace on those, so you know, it's just an assumption I would make. I agree that there's probably a lot of fermented foods that don't have characterized microbiological content, and therefore they could not be considered probiotic.
Does ISAPP have information about these specific species on the website that are considered probiotics?
That's a great question, because now I'm going to have to I'm going to have to tweak your question a little bit, because the first answer is no, but your question is important, because we really wouldn't talk about probiotic species. We would talk about probiotic strains, and the evidence for the most part is tied to those strains. Now, there's been talk about experts in the field trying to pull together a list of, you know, what they've called a positive list of probiotic strains. ISAPP has never been involved in that, and I've never actually seen a list. It may exist, but I've never seen one. But of course, a list like that is difficult, because you also have to recognize the context is that you have to make sure it's tied to the proper dose as well, and then if there are any matrix issues, and you know, you have to consider that as well.
So for producers, we had a few questions from producers asking, what's the best way to communicate the truth about probiotics to our customers? How do we responsibly discuss potential benefit of fermented products?
Right, and that I think is the key. So your product may meet the criteria of a probiotic, and if so, have at it, use the term and go for it. But if it doesn't, then I think what you have to do is say, well, how do I communicate the benefits of this product? And so first of all, just don't use the word probiotic if it doesn't fit. And then secondly, you can focus on nutritional value. You can focus on the presence of live microbes and speak to the association of those live microbes with health benefits without leading people to believe that in fact those benefits are confirmed. These are foods, and so nobody expects foods, or they're primarily being consumed for their nutritious components, their nutritional value. So a health benefit is kind of above and beyond, and so these can be great foods that are healthy, have healthy components, and then you may get live microbes that may provide some additional benefits along the lines of what Justin Sonnenberg would write about. And, you know, but you just have to communicate that in a way that doesn't lead people to believe that this is a probiotic that's been studied and shown to have a health benefit.
I love what you said earlier too, that consumers are becoming more savvy on the term live active cultures, that would be something more appropriate to put on a product, make a product claim rather than
Right.
probiotics. Okay, can you tell us a bit about para or postbiotics? Those are the new thing in the industry. Do you guys, do you study that at all?
Yeah, in fact, ISAPP is coming out with its probably last consensus panel paper on the topic of postbiotics. So the first recommendation I'll make is let's just get rid of the word parabiotics or paraprobiotics. We don't want to be dealing with that at all. Let's just coalesce around the term postbiotic. And, you know, very interestingly, especially from my point of view, where I've spent so many years focused on the live microorganism, which is a probiotic, you know, evidence is building that postbiotics, which are preparations that have been derived from live microorganisms and retain either intact or cell components of those live microorganisms, as well as all the other you know, fermentates and metabolites and other substances that are produced during the growth of that microorganism. And then those are being tested for health benefits. And lo and behold, they do have some. Excuse me. And so I think that this is a really interesting development. I would just advise you or encourage you to just keep an eye out over the, you know, sign up for the ISAPP newsletter, because as soon as this paper comes out, we will send out an alert to everybody, and you can read all about it in the paper from the consensus panel.
Great. All right, so this is a question probably for both of you, for both Mary Ellen and Maria. That was a fascinating hypothesis from the Sonnenbergs that we should be, you know, consuming live microbes. Do you think it is, let me pull up this question, sorry. Do you think we should associate that with live microbes or with their metabolites that could be associated with the health benefits of fermented foods?
Yeah, I mean, I'll take a stab, Maria, to start. I think that the live microbes are delivering way more than just metabolites. And so, yes, metabolites probably can be important, but I think that if you get the live microbes along with the metabolites, you're probably getting a lot more. And I think that these components can interact with different cells throughout the human body and have different physiological effects. And so I think both could be important.
Yeah, I don't disagree with that. I would say, you know, we have to use metrics, and what we can measure are live microbes. And then if we want to measure metabolites or understand the roles of certain compounds, that's interesting and deserves sort of a separate discussion. Keeping in mind that certain microbes in fermented foods are going to survive in the digestive tract, where the compounds that are in the foods may not make it to the intestine. So interesting question.
Mm-hmm. That's a good point.
Okay, so we had to follow up on that. We had a question: is colonization in the guts of the given strain still a criteria to define a probiotic?
No, and I'm going to say that so emphatically because first of all, there's very little evidence that any do. The vast majority of probiotics that have been tested are transient through the gut. They do not colonize, and that is a age old. I mean, that's from 25 years ago, 30 years ago. People said, well, it has to colonize, and there has never been evidence to substantiate colonization. Yet we have lots of papers that show that there's a health benefit. So clearly, colonization is not required, and it's never been a requirement for being a probiotic. If you look at the definition, it does not say that, you know, it does not speak to colonization.
Thank you so much. We had a lot of questions we didn't get to. I am going to put Mary Ellen's email in our chat bar right now. So if you have additional questions, she has graciously offered to answer them. Again, big thanks to Dr. Sanders and Dr. Marco. Thanks to all of you for attending today's webinar. We will post a recording on TFA's website in the next 24 hours. We also have a number of great webinars coming up in the next few weeks, including the new definition of fermented food and the many sides of kefir. please go to fermentationassociation.org to check these out and register. And while you're there, subscribe to our YouTube channel. Thanks so much.
Bye-bye. Thank you.
Thank you.